Good morning, ma'am.
Good morning, ma'am.
Good morning, madam. Madam, streaming, madam. Yes, madam, it's working. Yes, madam, it's working.
Yes.
streaming is there.
Good morning.
Hello. Good
Good
morning,
morning.
sir.
Good morning. Good morning, everyone.
Uh, Principal, ma'am. Good morning, ma'am. Good morning, Tarun, sir.
Good morning.
Thank you for joining us today.
Thank you. Thank you for organizing this
Thank you,
webinar.
sir.
So I'm audible. My voice is coming clearly.
Yes, sir.
Yes, sir.
It's coming clearly, sir.
Okay.
Manohar, sir. Good morning, sir. Ravikant, sir. Good morning, sir.
Good morning, Lekhya.
Good morning, ma'am.
I think it was on mute, ma. That's why we were not able to connect.
Yes, ma'am. I am making everybody co-host, ma'am.
Yeah.
Thank you, ma'am.
Good morning, sir. Good
Good morning,
morning.
madam.
In fact, good evening, sir. Good evening for you staying back so late for
Yeah,
us. Thank you so much. Thank you so much for all your consideration of our webinar and being a resource
my
person.
pleasure.
We are
My pleasure.
extremely honored by your presence, sir. Thank you so much.
Thank you. Thank you for inviting me.
Thank you, sir. Thank you.
Hi, Chandana.
Ma'am.
Please start, ma'am.
Okay. Honorable Chairman of Sri Vidyaniketan Educational Institutions, Padma Shri Dr. M. Mohan Babu sir, our dynamic and revered CEO, Mr. Vishnu Manchu sir, Principal of Sri Vidyaniketan Degree College, Dr. D. Asha Lata ma'am, teachers and delegates of today's program. A very delightful morning to one and all. I am Sai Chandana Venkatila from second year BSc Microbiology, Sri Vidyaniketan Degree College, Tirupati. On behalf of Department of Biological Sciences, I feel privileged to welcome you all for the international webinar on recent advances in infection and immunity. I humbly request our respected principal, ma'am, Dr. D. Asha Lata, ma'am, to give the welcome address. Please, ma'am.
Yeah, good morning, everybody. We are in fact very happy that all of you have joined us. And it is indeed a great kind of a pleasure that today we are having an international webinar on recent advances in infection and immunity. We are very much thankful to Dr. Tarun Keshwani sir, who has joined us from US. Indeed, it is his, excellent interest in the field of research and the areas of evolving sciences of biological sciences that today we are here to discuss. And I hope of all the children who are part of this webinar will be very happy, in order to learn about the recent advances. Because in the series of webinars that we have been doing here in our college, Sri Vidyaniketan Degree College, we are focused on the recent things which actually open up research avenues for our students. So it is very much necessary that the advances in the infection and immunity, they become part and parcel of our life nowadays. Because as we look at the natural scenario as of now, it has been proven once again that those who have got the natural ability, immunity to withstand, whatever is happening across the world, the infections as a part of it, they have been doing excellently well. And their resistance to the diseases is quite good. So infections, yes, because of the viruses which have been on trail now. Like, there is a lot of, see, but once there was a time where there was no migration possibly done frequently, and there was no travel which was, cross continental once in a while. But as we now see a global village in the entire world, when all of us are interacting with each other physically, then I think, the infections do spread, and there is a kind of a cross contamination happening everywhere. Cross infections are the transportation of the virus is happening, and it is across the world. The spreading is very quick. It is possible in all ways, which is both psychological or physical, whatever it is. And because of the food that has been, part and parcel of the import and export systems, because where we are sharing whatever is the produce across the world, there has been a lot of changes in the ways that we are trying to become resistant to the organisms or the creatures. But, in fact, because of the chemical substances which are being used in the natural food production, or maybe because of the different kinds of the evolving sciences, maybe the resistance to the disease has become a part and parcel of not only our agriculture but also our general medication system. So people like Tarun Keshwani sir, they are so very good that they have taken up this noble profession of doing research, and especially on the core areas where human, civilization needs a kind of intervention to understand that, the part and parcel of our, life is these infections. But then how are you going to actually develop the immunity? How are you going to make your body more stronger? So biologically, when you see the anatomy of the body, like, it can resist. And like, recently we had a webinar on RNAs, like, natural therapy, which is done through the help of the nanobiotechnology or the cancer biology advances. So the part of our body which is there, which it can become immune to infections, is a new dimension, along with the scientific technology which can be used right now. So I think as a part of the most dynamic planet in this galaxy or this universe, Earth needs all of us to be more advanced in our thought process to make ourselves very strong, and in fact very progressive, to think about the latest research and advance. Thank you, Tarun sir, once again for being with us here today. We are very much indebted and thankful to our very distinguished and honorable chairman, Sir Mohan Babu sir, Padma Shri Dr. Mohan Babu sir, and also our very dynamic CEO, Vishnu sir, for giving us this opportunity to host this webinar today. So I'm very thankful and sincerely congratulate the organizing team members, my faculty team of students, and the faculty members and the staff for making this a kind of a happening today, a reality today. I hope that we'll get the best of the knowledge to make ourselves more immune towards the infectious diseases and fight it out and make ourselves the best of the God's creation. Thank you, one and all. Have a wonderful session. God bless all of us.
Thank you, ma'am. Now I request our webinar coordinator, Dr. Charles Lekhya Priya, ma'am, Assistant Professor, to introduce today's eminent resource person. Over to you, ma'am.
Thank you, Chandana. A very good morning to one and all. It gives me great pleasure to introduce and welcome the resource person of today's international webinar, Dr. Tarun Keshwani, sir. Dr. Tarun Keshwani is currently working as postdoctoral scientist, Albert Einstein College of Medicine, New York, USA. In 2019, he worked as a senior postdoctoral research fellow in the Institute of Molecular and Cellular Pharmacology, France. During this period, he worked extensively on the immunogenicity of Ebola vaccines and their mechanisms of action in France, Sofia, and Tripoli. He also worked in the Institut Pasteur Lille, France, as postdoctoral research fellow from June 2016 to Jan 2019. In this time, Dr. Tarun worked on the development of new approaches to understand the complexity of infectious diseases. Dr. Tarun received Doctor of Philosophy in Parasite Immunology from University of Kolkata, India. Dr. Tarun has sound knowledge in the concepts of infectious disease and toxicological studies, animal handling, and Ebola immunogenicity studies. He is proficient in panel design, flow cytometry, cell sorting, in vivo imaging, image stream analysis, MRI data analysis, and interpretation. Other skills include mammalian cell culture, biochemical assays, immunological assays such as ELISA, Western blot, immunofluorescence, and immunohistochemistry. Dr. Tarun is well experienced in grant writings and successfully defended scientific projects. Dr. Tarun has published several research papers in the journals of high repute. He worked as an internship tutor and guided students to complete their projects in infection and immunity at University of Lille, France. He worked as a supervisor and guided several student projects in the areas of molecular and cellular biology, parasitology, and mycology at Sorbonne University, France. He is a fellow member of American Society of Tropical Medicine and Hygiene, French Society of Cell Biology, Indian Immunology Society, Indian Society for Parasitology, and QSA University of Kalyani. Dr. Tarun has a big list of awards to his credit. Fellowships and awards of Dr. Tarun Keshwani sir are as follows. Young Researcher Prize in Neurobiology from Foundation of Trials, France. Gold Medal Professor M.B. Mirza Award for contribution in the field of parasitology at 26th National Congress of Parasitology, India. Raman Chapak Fellowship from DST, India. M.M. Chakrabarti Memorial Award for contribution in parasitology awarded by Zoological Society, Kolkata, India. Young Scientist Award at Society for Free Radical Research, India. Elsewhere Prize for contribution in human parasitology at 24th International Conference, Australia. International Travel Grant from Department of Science and Technology, India. Senior Research Fellow, Junior Research Fellowship from Research Fellowship in Science for meritorious students to promote quality research, UGC, India. Sir, despite your busy schedule, you have accepted to be the resource person, and on behalf of the organizing committee, I thank you and welcome you for today's session. Thank you, sir.
Thank you, ma'am.
Thank you. Thank you. Yeah.
I deem it an honor to invite today's esteemed resource person, Dr. Tarun Keshwani, to enlighten us about the recent advances in infection and immunity. All the participants are eagerly waiting to listen to you, sir.
Uh, Thank you very much for the nice introduction. I mean, I've also forgot a lot of things which you introduced today, and thank you, Principal Ma'am, and all the organizers. Uh, it's a great honor and privilege to be part of this international webinar. And now I would love to share my slides. Okay. So thank you again for the nice introduction and organizing such an international webinar and the thought process behind this. I should thank all the people and my friends who helped me to connect this, and Priya also for connecting this part. And the main motto of this is to help students to pursue research, which I understood while discussing with Principal Ma'am and the other organizers. So I will try to make it simple and very lucid, so that even with the current pandemic situations, we are aware of a lot of facts and figures in infection and immunity and the recent advances. But I would love to present in a way so that it will be clear. The message should be clear to the masses. I mean, because there are a lot of information floating around this time at this point of time in the world. So I would again love to thank Sri Vidyaniketan Degree College, Department of Biological Sciences, and all the organizers and all the faculty, students, scholars, and friends participating in this webinar. So I'm, as I was introduced, that I'm working as a postdoctoral scientist at Albert Einstein College of Medicine from this year, beginning of this year. And here also I'm working primarily on the coma and cerebral malaria aspects. So I will distribute my presentation in four aspects mainly. That is a fundamental of infection and immunity. Then it will be on the current trend, that is coronavirus pandemic, COVID-19. Immune response triggered by vaccination, as Professor Ma'am already mentioned, how we are generating immune response, and with the transportation and all the cross infections happening. So I will elaborate a little bit on that part. And the basic and translational human immunology, which will be more helpful for the students, that how the translational research works. So the fundamentals of infection and immunity. So what's infection disease? So the infectious disease is the presence of particular microorganisms in part of the body where it is not usually found. So what happens when a particular microorganism is found in a body where it shouldn't be found? If it overcomes a body's defense, the state of disease results. And the pathogens is a disease causing microorganisms. We are all aware of this aspect from our school days in biological classes, but now it's hype. We know that pathogenic microorganisms have special properties that allow them to invade of the human body or produce toxins. With the current pandemic, the special properties are super special. I mean, we are still working on that. Scientists globally are still trying to understand the properties behind the coronavirus. And host, we are the host in this case, is an organism that shelters and supports the growth of the pathogens. So sometimes we are able to protect us from the foreign pathogen, and sometimes it's detrimental, we are unable to. So how do different antigens or pathogens direct different dendritic cell? So how do different antigens or pathogens direct different DC maturation programs? We know there are various pathogens like viruses, bacteria, fungi, parasites, which presents the antigen to dendritic cells. The dendritic cells then engulfs the antigen. It presents the antigens to the naive T cells, and which can be differentiated to CD8 T cell, the cytotoxic T cell, which can lead to the Th1 cell help, and then to the granzyme and perforins, which can be mediated by intracellular bacteria, viruses, or protozoa. T cells can also be differentiated to CD4 T cells. The dendritic cells can interact, and it can lead to Th1 cell, Th2 cell, Th17, or Tregs. So it depends on the microenvironment and the cytokine levels, which can help the CD4 T cell to differentiate into Th1, Th2, Th17, or Treg. On the other hand, the T cell can also help, which we are listening more these days, the antibodies to help the B cell to activate, and the cytokines generated by T cells helps the B cells to activate, and which leads to plasma cell, which generate antibodies. I will explain that in details later on. So this is a in general concept how the immune system works, and in which direction the immune system will lead depending upon the pathogens we are invaded with and the organ where the pathogen has invaded and the cell types in the local microenvironment. So it's a complex system. So the fundamentals of immunity is that due to current knowledge of human genome, animal genome, and several imaging techniques, application-based studies, clinical trials led us to understand the immune regulation and the immune response undergoing during an infection. There are several environmental factors which I just mentioned, that the viruses, the bacteria, the allergic food, vaccinations. All this helped us to understand the fundamentals of immunity, how it regulates, how the myeloid-derived suppressor cell works, how the B cell works, and how the immunity develops through pathogenic CD4, CD8 T cells, the innate activatory receptors, the cytokine response. So overall, this cycle of immune regulation till date we had understood in depth because of the several clinical studies, animal studies, translational studies. So we have a background of information that what is going on, what is the basic of immune response. So this is an example of the immune checkpoints in being targeted by therapeutics for cancer, infectious disease, or autoimmunity, where these checkpoint molecules, which has a biological role and the therapeutic names, the disease where it has been applied, their respective roles. So we know we have a bunch of informations which helps us to reveal or understand newer diseases. In cancer, in infectious disease, we has progressed to a greater extent, but still there are a lot of new immune cell markers, mechanisms are undergoing. We are getting new informations day by day. So on the other hand, as I was speaking about the pathogens helps us to exploit uh, the new ecological niches. So if we see the infection outcome is dependent on the complex and multiple interactions between host immune mechanisms, pathogen, and environment. In this case, we can see that the environment is an important factor. The health, geographical locations of each individual, if we compare United States and India, it's different. The food habits, everything is different. The viral strains are different because we have seen there are mutations in viral strains. The immune response towards the chronic infections, asymptomatic carriers, reservoirs for viral transmissions, impacts the multi-ethnic populations, the susceptible hosts, the environment, the pathogen, which finally leads to the disease outcome. And this changes in the ecology and biodiversity, which happened where the pathogen outbreak occurred, is also an important part in this disease triangle. So moving forward, the major emerging and re-emerging infectious disease outbreak in last 2 decades, if we see that the SARS outbreak did happen in the year 2002, then also the MERS, which we are speaking about very frequently these days, it also occurred in 2012, and now the COVID-19. In between, the Ebola outbreak occurred in 2014 in Democratic Republic of Congo. The Zika virus outbreak occurred in 2006 and again in 2013. So all these re-emerging infections outbreaks occurred because of the communication, the movement of the infections pathogen, the change in the microenvironment, which further makes us to understand this disease triangle is very important to understand the future pandemics, and each reports, each study help us to understand the new pandemics coming up or new outbreak of disease happening. So research, multidisciplinary research and the translational research is a very important aspect, especially the immune response and studying the infections and immunity is very important to understand the disease mechanism. So as I was mentioning, the origin of SARS-CoV-2, as we know, originated from pangolins, but it mainly originated from bat. As we have seen during the 2002 outbreak and 2014 outbreak, it originated from camel and the civet. But in this case, we still are not sure how the COVID-2 transmitted to human from pangolins if they really did. So there are still studies going on this aspect, and also the studies going on the genome of the SARS-CoV-2. So now the coronavirus pandemic, I mean, it's almost we have crossed the half of this year, 2020, but still we are in huge stress as we never went through in last 2 decades regarding any pandemics in our lifetime. So this is a very important part, and just would like to explain the symptoms as we all are aware of with this current uh, mass communication systems. So it causes the high fever and pneumonia, which is very common with similar with the common cold or flu. In USA also the flu is very common. So initially people are confused whether they have COVID or flu. The primary symptoms are fever, runny nose, dry cough, shortness of breath, fatigue, body aches. And the transmission, as I mentioned, initially the transmission, now what people are understanding that spread from person to person through sneezing or coughing. I mean, that was the main factor why there was huge outbreak. And the respiratory droplets containing the virus can remain in the surface even after the ill person is no longer near. So this part is still under research, that how far and how quickly the virus can transmit. So this was study from University of Alabama and Birmingham. So till yesterday, the active cases in India, if you can see that that there are almost 6 million cases in mild condition and serious or critical, which is good news at 1%, but still it's a huge number. And in closed cases, we can see that there were almost 5% deaths, and recovered or discharged is a huge number. And here, if you see the red line, which is the deaths and the active cases, and the green one is the recovery. So if we understand the phases of response to disaster, because in the beginning of this year, we had a warning. Each of the countries had a warning. And the impact to that warning is very important, how the impact was taken seriously by the respective authorities, and how the cases reached to the peak phase. And with respect to time and weeks after it subsided down, depending upon the infrastructures of each authorities and the measures taken by each individuals at individual level, the cases again get rise. This is general approach, and the second disaster comes after. So the enhanced community and individual adaptation occurred there, which is now termed as herd immunity sort of, and the previous level of equilibrium reaches up. So if you understand the modeling infectious disease dynamics, which now with this current pandemic, people are understanding more, or they are more aware of the term, that is herd immunity. So how this herd immunity is reached? So this is a normal fraction infected, and the herd immunity concept is based on the R0, which is the expected number of cases caused by an index case. So this modeling of infectious disease dynamics is still under consideration. There many countries tried this aspect, but some failed and somewhere it worked. So as this disease is really new, it's very hard to understand. So this was a general concept in a recent paper in June, where they are trying to explain the different outcome to each individuals and the monogenic causes of COVID-19 susceptibility or resistance. So if we consider here, all dark individuals are uninfected, and with the spread of SARS-CoV-2 infection, with the age and comorbidities, means as more the comorbidities are, and as the age increases, we can see the red individuals are prone to severe disease. On the other hand, this lower half, they are more asymptomatic, the blue. Among this, also the one rare idiopathic severe disease in the young, which is called the monogenic cause. So this individual, even in the range where it has lower comorbidity aspect and age is also less, there are still few cases where they have a severe disease. On the other hand, in this part, where all these individuals are more prone, but there are few individuals which are rare, unexplained, resistant to the infection, and most of them here are symptomatic. The blue ones, the purple ones, and the light blue are asymptomatic. So this is a way to explain to how the human genetics of protective immunity to SARS-CoV infection works or working. So still, the scientists are trying to understand this aspect, why there is a difference and in rare, unexplained, and idiopathic diseases in young and adults. So, by Ruth Caron, she's a vaccine researcher at Johns Hopkins, and it was mentioned that I think this virus is with us to the future. On the other hand, but so influenza with us, and for the most part, flu doesn't shut down our societies; we manage it. But here we are unable to understand in greater extent what consequences it might cause, because it might have some after effects which scientists are studying worldwide. So understanding, or as I was mentioning before, to here the knowledge of flu, previous knowledge of flu is helping the scientists to understand the current pandemic situation in a much better way, in a quick way. But time is too short, but things are to be done in a greater extent. So there are some common mistakes and misconceptions that I would like to mention that not all microbes are pathogens. Some diseases have been nearly eliminated through the use of vaccines. And some people may think that vaccines provide permanent immunity to a disease, but it's not the case for the flu, normal flu. Because every year there are almost mutations in the flu viruses, and so the shots are repeated with the modulations. So we have to understand that vaccines are, in some cases, are not permanent, and it doesn't provide permanent immunity to a disease. So the concept of herd immunity, we have understand, we are listening everywhere in articles, in news channels. In this scenario, we can see that there is no herd immunity. So the blue ones individuals are susceptible, and the red individuals are infected. So the herd immunity concept arrives after 70 to 90% approximately individuals are infected, and then the herd immunity achieved. Then here the green ones are immune. But this is not always true, because this is a new aspect, and still scientists are working hard to understand the true disease transmission aspects. But still, this is the basic concept, and how this herd immunity can help slow or stop the spread of the disease. So the coronavirus vaccine tracker, everybody is eyeing on this aspect these days, as vaccine typically require years of research and testing before reaching the clinic. But scientists are racing to produce a safe and effective coronavirus vaccine by next year. Fingers crossed. I hope everything will work well, and we will soon be in a sigh of relief. So currently in phase 1, there are almost 23 vaccines. In phase 2, 14, in expanded safety trial phase. In phase 3, in large-scale efficacy tests, there are 9. By early January this year, and the first vaccine safety trials in humans started March, but the road ahead remains uncertain. Some trials will fail, and others may end without a clear result. But if you may succeed in stimulating the immune system to produce effective antibodies against the virus. So, as I was mentioning, that the research in the flu, in the previous SARS-CoVs, helped this time to understand the coronavirus 2, because in 2002 the SARS-CoV entered. It was studied that it enters cells through host cell surface receptor angiotensin converting enzyme 2 ACE2. We can see that it enters here. And in recent research suggests that the SARS-CoV-2 cell entry is mediated through the same pathway. So these findings demonstrate a strategy to identify. This is an example how the research is being translated. So these findings demonstrate a strategy to identify conserved antibody binding sites on SARS-CoV-2 glycoprotein, which can be subsequently targeted in vaccine design. So the vaccine testing process, after designing, there is a testing process. So there are several phases of vaccine testing, as I was mentioning before, and
Sorry to interrupt you, sir.
Yeah, sure.
Sir, actually, I think your slides are not moving.
Not moving?
Yeah, it is stuck at coronavirus vaccine tracker.
Okay. Thank you for informing me. So I will stop sharing, and now it's okay?
Not it. I think it is hanging there now.
Okay, no problem. Just give me 1 minute. I will. Thank you for the patience. Is it okay now?
Yeah.
Is it moving? Okay.
Yeah.
So I will again start from this part. I hope it will be okay. Yeah. So, yeah, can you repeat, please?
Yeah, I think previous slide was the one where it got.
Yeah, this one.
Yeah.
Yeah, yeah.
Now it is fine.
So I will, yeah, thank you very much. So as we were discussing about this, the vaccine, so the SARS-CoV-2 genome studied helps us to understand, and the study, the work began from January. The first vaccine safety trial started in March, but the road ahead remains uncertain. So what I was trying to explain that some trials may fail and others may end without a clear result. But a few may succeed in stimulating the immune system to produce effective antibodies against the virus. So moving from the part that the translational research is very important, and the information from the prior cases, prior outbreaks are very important. And here also there is an example that in the 2002 SARS-CoV-2, when it was studied that enters cells through host cell surface receptor angiotensin converting enzyme 2, ACE2. And recent research suggests that the SARS-CoV-2 cell entry is mediated through the same pathway. So these findings demonstrate a strategy to identify the conserved antibody binding site on SARS-CoV-2 glycoproteins, which can be subsequently targeted in vaccine design. So this is an example of one vaccine design. So previous studies helped the current study to understand, and that is in part where the previous research helps to understand where a new science or scientific subject can move ahead, which will help students to develop a new projects. So the vaccine testing process, in the preclinical testing, the scientists test a new vaccine on cells and then give it to animals, such as mice or monkeys, which I will explain later on. The importance of animals in translational research from human to uh, mice or monkeys. And so there are confirmed 89 preclinical vaccine in active development. So they are in phase 1, phase 2, phase 3 efficacy trials, early or limited approval, combined phases, and approval, which I explained before. So what's phase 1? Phase 1 has limited subject, 5 to 50. It assesses safety, immunogenicity, and the dose scheduling conducted in individuals at low risk for infection. Phase 2, expand safely, immunogenicity, and optimize dose schedule may include a risk population, usually double-blinded, placebo-controlled, and randomized. So to assess the safety of the vaccine, phase 3 is very important, where we have higher individuals, 100 to 10,000, and it's conducted in populations specifically at a risk for the infection. So this is very critical phase for the vaccine development. And after this phase, the phase 4, it assesses the safety and effectiveness under item conditions and detects rare adverse events. So there are methodological issues and problems. The vaccine trials may take months or years to complete, since a sufficient time period must elapse for the subjects to react to the vaccine and develop the required antibodies. It will depend also on the vaccine strategies, how much beneficial antibodies it will be generated and long-lived, which I will come later on. So how the immune response triggered by vaccination. So the immune response triggered by vaccination depends on several aspects. The first one is the formulation or the drug formulation, the delivery system, and the adjuvant, the antigen dose, and the route of priming. So what is priming? A priming helps to understand, it boosts the priming from the vaccine when it enters the system, the area where the vaccine has been adjuvanted. The dendritic cell takes the antigen and it primes the CD4 T cell. The CD4 T cell then differentiate, which I explained in the beginning, that to the several Th1, Th2, Th17, Treg cells, depending upon the cytokine microenvironment, which may lead to differentiation in affected T cells. T cells also helps, the T follicular cells also helps in the germinal cell reactions, which produce long-lived plasma cells, and also the short-lived plasma cells are produced. So what's long-lived and short-lived? The short-lived means they stay for for very long time, and long-lived, they stay for long, and they have higher protection capacity. So this antibodies, after they generated, they can be detected in the plasma, in the blood, which people are studying now, the antibodies generated against the COVID or other diseases people who study. So then this antibodies are also, if it's boosted, if there is a second infection, reinfection, then it generates the memory response if there is, and this B and T memory cells in the lymph nodes are very important for the protection. Now in this B and T memory cells, if the memory cells are long-lived or short-lived, depending upon that, the efficacy of a vaccine is determined. So this takes several months or years to understand which vaccine formulated is more efficient and better long-lasting uh, potential. Like in flu virus, I explained that there are mutations, so the memory response doesn't work. So that's why there are new vaccines for that every year, new shots. So if you understand in the context of COVID till now, the immune response, inflammation, and the clinical spectrum of COVID-19, if there is no infection, there is protective immunity that suggests. If you see this here, we have the broader aspect, the green one, neutralizing antibodies, cytotoxic CD8 T cells, Th1 cells. And if there is infection, symptomatic, mild, so that is still on this part, means protective, moderate, so there is less, but there is chance of getting towards immune dysregulation. And if it's severe, it will lead to death or recovery. If recovery, then that means it's going toward the protective immunity. That's why recovery. And if it's death, that can be due to acute inflammation, cytokine storm, acute lung injury, coagulopathy, and multiple organ system dysfunction. But here is another factor, that is we still don't know what is the after effect of this, and the antibody, if generated, how long it will give the protection. So there are a lot of research to be done yet. Scientists globally are working on this aspects, and they are really working very fast in team, and that shows the collaboration, importance of collaboration in research, and from field to bench. This is very important aspect. So as I was mentioning, the memory cell response is very important. With the first exposure of virus, the response expanded here depending upon the population size, and this is the days post-infection. Then with the second contraction phase, cells die over next several weeks, the CD8 T cells, and the memory cells subsides down. With the subsequent infection, the memory cell longevity is very important, and the pool of the affected T cells can be separated into multiple diverse subsets based on difference in gene and protein expression, effector functions, the migratory patterns to the site of infection, proliferative capacity, and long-term fate. However, the other T cell subsets with intermediate differentiation states also exist that have mixed phenotypes. So longevities and abilities to self-renew are depicted by here. If you see that the more memory T cells, and they should have a longevity. So this red bar, if you see, there are more long-lived memory T cells. The long-lived means the longer the protection. So why I'm mentioning this memory T cell part, because now it will be very important after the vaccine design that whether they have a memory capacity or not. But in case of, in general aspect, in case when a vaccine is designed, this is studied that the memory T cell subset should be able to produce high level of IL-2, that is SCM means stem cell memory, which is central memory and effector memory. So if there are more stem cell memory or central memory producing IL-2 cytokine, is subtle amount of IFN gamma and less cytotoxicity, it means that they have a better protection capacity and memory response. So these are some immunological markers, transcription factors, which helps to define between short-lived, long-lived, and the memory precursor molecules. These are the graded activity of transcriptional programs. And in various vaccine design, scientists do keep in mind, and still there are new transcription factors, markers coming up, which helps to better understand the design. So this is always not possible to study in human or with the plasma samples from human or the cohorts. So that's why the translational research is very important, where the research is conducted on the several types of models, animals, primates, in vitro research, where the computational system comes. So then comes the aspect of the basic and translational human immunology. What's the importance of that, and how, with the current situation, it will lead us to a better resolution. So there are advanced model systems and tools for basic and translational human immunology. We can do blood-based immunoassays, reporter analysis for antibodies and titers, TCRs, modeling immune tissues, tissue-based immunity study we can do, and organoid-like culture. Organoid-like culture means we can grow organs in culture. There are several studies going on, like hepatic culture, brain culture. It will behave like an organ. So from human to in vivo, that we can do that to understand the underlying mechanism. There are also in vivo studies which help us to understand the natural immune variation in silico models and bioinformatics. These days, the bioinformatics and in silico models approach helped to a greater extent to understand the current pandemic situation and the COVID-related genome studies and other, which will definitely help in the vaccine development, at least to some extent. So the wealth of human data for translation immunology is of great importance in cohorts, in patients. With now also, if we go through several science fictions, newspapers, that several research are going on with the COVID, where patients who were infected and who are recovering, their samples, the scientists are working on, and the research is going on. So there can be various kind of specimens from cohorts. Their tissue sample from the damaged tissue sample, the specimens, the urine samples, the stool samples, from where the cell specificity can be studied, the BDJ recombination can be studied. From flow cytometry, we can understand from the plasma cells, the PBMCs, what is the function of the immune cell types in the disease patient compared to the control patients. We can study the cytotoxic issues, the functional aspects, in vitro studies we can do. We can also understand the environment, that is the pathogens or the virions, what's their role. We can understand what's the fate of that. So it's very important to understand the disease mechanism. So an overview of the animal models for translation studies for human immunology, as I was mentioning before. So there are several model systems which help us to understand in a conventional way. Like mouse models, we know the advantage is consistent, widely available worldwide. We are aware that we can work in mouse models and is easy to handle, but it some way poorly represents many human diseases. Not all diseases are directly related or or can be modeled in mouse models. So it helps us to understand the preclinical work in some cases. Translatability is less, so the as more we go down, it will increase. Then is the next generation mice, that is humanized mice, where in the mice model we can grow specific human cell or engineer tissue, which are more potential for translation to humans. Many immunologic reagents easy to handle, but they are more expensive than the conventional mouse model. Here, the single organ infections can be done, such as liver. Only the liver uh, will be implanted in a mice in such a way that it will totally uh, replicate what is going on in the human system, which sometimes it's not possible or ethically correct to do in the humans. So that's why we need to study on the model system. So their translatability is higher than the conventional mouse, than the non-human primates. But non-human primates are mainly very similar to the human-like model, but there are ethical concerns. It's not always easy to understand that, and also the MSC are larger than the humans. There are studies on non-human primates, on HIV, tuberculosis, many arthropod-borne disease, but there are also some studies on tuberculosis which went very bad when it was translated to humans after doing the research on non-human primates. So it should be taken into consideration that it can be risky also. There are other animal models, but the reagent limited or non-existent, like ferrets. Many as typical are not bred for research. But basically, this model help us to understand the human disease mechanism. So, what is the future of this translation human immunology? I will say it's bright because in the past we were mainly translating human immunology from animals.
Hello, Varun sir.
Madam Sir got disconnected, I think.
I'll
Ye-
call him, sir.
Yes, there is some problem, it seems, with his net connection.
Yeah, yeah.
Hello.
Yes, sir.
Yeah, it's okay.
Yes, sir. Now you are visible, sir.
So, from where did it start? Can you tell me? Hello.
Tables, sir. The
Tables.
table you were explaining, yeah.
Yeah.
Yeah. This
So, yeah, I'm extremely sorry for that. I don't know why it happened. So, I will explain again this table. So, as I was explaining, that the animal models for translational studies for human immunology and its importance. We know that there are several models people are using, like conventional mouse models, next generation mouse, humanized genetic or tissue engineered mouse models, non-human primates, and other animal models. So each of this model system has their own pros and cons. So starting from this conventional inbred wild type and knockout mice, they are easily available compared to the next generation mice. But the inbred mice poorly represents many human diseases. On the other hand, they are less expensive than the next generation mice models, but each model give us or help us to understand individual diseases. But on the other hand, non-human primates, they act like human-like model. There are many immunologic reagents, but there are ethical concerns and hugely expensive. And the laboratories for non-human primates are not always easy to get approved. So there are many studies on has been done on non-human primates on HIV, tuberculosis, and many arthropod-borne viruses. But there are some risk factors also. So what's the future of translation human immunology? So as I mentioned, there was a technical glitch also. So I will explain again that in the past, the basic discovery of disease study and treatment in mice was mainly occurred, and from that it was validated in NHPs, that is non-human primates, and then testing in humans. But things are changing now. In present, we are gathering sample or data from humans, confirming in animal models, non-human primates, in vitro systems, and correlating and predicting with the bioinformatics. But in future, the mass cohort collection from numerous relevant sources will help us to understand, and there will be less and less usage of the animals, but they will be needed, but there will be lesser usage of animals, non-human primates, and more will be the ex vivo and the computational analysis in vitro models. So in summary, the researchers have been rummaging through results from the common cold study and others like it, while scrambling to understand the human immune response to SARS-CoV-2 using animals, cell cultures, along with the latest molecular and immunological techniques. The antibodies, the design vaccines and therapies using animal studies are also going on, but there is no quick and simple experiment that can firmly determine whether immunity will be effective or lasting. So that's why there are several aspects, like I mentioned, the memory response, the antibody response, long-lived. So it's just too soon to know what's going to happen. So what the immune response to coronavirus says about the prospect for a vaccine. I will say that the immunologists are on the hunt for the antibodies, B cells, the T cells that help to kill SARS-CoV-2 in the body. So, how much time I do have more?
You could take your own time, sir. There is no problem.
Okay,
Thank you,
thank
sir.
you. So, this is one important aspect which I would like to mention, that the emotional health during the COVID-19 pandemic. So as we were going through the COVID-19, the immune response, infectious disease, but there is also a psychological aspect where the psychiatry and the virtual support comes into. So scientists worldwide are working very hard to support mentally, emotionally, to because this is a stress situation, as I mentioned in the beginning, which we didn't face in last 2 decades, as we faced this year didn't pass yet. So this virtual support center is a very important aspect which supports the emotional well-being, health and issues, child care, several aspects. Because if this is provided to a greater extent, that will also help the other people who are into research, into essential aspects, so that they can focus on their work also. So this is one aspect which I thought I would like to share. On the other hand, we know that in last few months there are a lot of aspects that malaria is an can be an important factor. Malaria medicines can be an important factor. I'm not going into that, but there is a recent report that the malaria could have a resurgence due to COVID-19. I mean, the potential impact of health service disruptions on the burden of malaria are worse. And if things goes like this, where the other diseases will be neglected due to several reasons, then African children might have enhanced malaria cases by end of this year, which will be approximately 70% among children under the age of 5. So this should be taken under consideration, and I am sure that the authorities are working on that. So a little bit of malaria background, as we were discussing about the infectious disease and immunity. So I would like to add a parasite aspect also. This is my forte. So this is a vector-borne infectious disease and caused by protozoan parasite of the genus Plasmodium. And according to the disease severity from rare to mild, this is the more severe and this is the less severe. Falciparum causes more deaths worldwide with the cerebral malaria, and according to WHO, the definition is coma and the peripheral parasitemia. From that, we understand that there is cerebral malaria. So this is life cycle of malaria. Mostly all of us studied in school and colleges. This is in two phase. Here the mosquito phase and here the human phase. And so the mosquito injects porocytes via bite, and it goes through several phases. After mitotic replication, it comes out from the liver and ruptures the merozoites. The merozoite goes to the gametocyte phase, and also here it goes to the asexual asymptomatic cycle. So this is the phase where we are able to know that we are into malaria after doing the peripheral staining, or if someone has a severe stuff where it shows the symptom. But otherwise, this is asymptomatic phase where in most cases, and there are cases that it has been known that the parasite stays in the liver in a dormant stage and it comes relapses after. And then the mosquito again, if bites the infected person, and it takes the gametocyte, and this cycle continues like this. And this is the phase where we can understand the symptoms. So 68% of the global malaria deaths occur in 10 highest burden countries and estimated 228 million cases worldwide in 2018. Among that, there are almost 405,000 deaths. Unfortunately, 70% of malaria deaths are children under 5 years old, and from malaria in Africa, one child dies every 2 months. With cerebral malaria, respiratory distress, metabolic acidosis, and severe. So symptoms of malaria, as we are all aware of it, as headache, systemic fever, back pain, spleen enlargement, respiratory dry cough. Nearly all malaria deaths result from infection Plasmodium falciparum, as I mentioned before, and multi-organ failure. So here I'm trying to explain how translational research helps and what's the need of the model in context of protozoan infections and the role of immune response and the knowledge which help us to understand the disease in a better way. So in malaria, in the cerebral malaria, as I was mentioning, so this is a blood-brain barrier, and this is the lymphocyte, monocytes, the neutrophils, and this is the astrocytes and the microglia. So during severe malaria, this blood-brain barrier disrupts, and there is infiltration of the cells in the brain, sequestration in the central nervous system, and the disruption of the blood-brain barrier, acute inflammation. The CD8 T cells play an important role. There is activation of glial cells and the cytokine burst in the acute infection. So what happens here in the mechanism of cerebral malaria? And all these years of research help us to understand the mechanism going on during cerebral malaria, but still there were almost 26 vaccine candidate tested in the field, and the artemether is one of that, and RTSS, but not all the vaccines provide 100% efficacy. So there are still a lot of research to be done. And in the brain, when the parasite enters the blood-brain barrier, it disrupts here. It forms the clumping, autoagglutination, the infected RBCs. It forms a rosetting. It adheres with the endothelial cell linings, which obstructs the blood flow, leads to the hypoxia and altered blood flow, means reduced oxygen levels, which releases the toxins, which further leads the recruitment of leukocytes and platelets, and it causes a cytokine burst, which leads to the breakdown of the blood brain. And then the question was, which I was studying, that whether the B cells or antibodies playing any role here or not. So theories about pathogenesis of cerebral malaria: it causes impaired cerebral blood flow, sequestration of infected red blood cells in blood vessels. As I was mentioning before, the blood stage, the asexual symptomatic phase where the RBCs gets infected, that transfers to the brain blood, and it sequesters there, which leads to the hypoxia, and it causes edema in the brain, which leads to coma. So the generalized immune response is the high pro-inflammatory response, low anti-inflammatory levels, and animal experiments help us to understand these aspects. Because the key questions still are: what is the pathophysiology of severe disease? How do people living in malaria endemic regions become protected from severe disease? And all this cannot be answered fully by observations of infected humans. So the Plasmodium parasite and the host immune system maintains a tenuous balance, because the parasite stays in the host, in the human beings, and this cycle is continuing for centuries. Mosquito is transmitting the parasite to humans, and from humans, then the mosquito is again taken. So this parasite and host immune system is complicated. So difficult to unravel the mechanisms of protection or pathology in humans, because not all human beings are going through severe malaria, coma, or deaths. We do get cured from malaria. On the other hand, in African countries, there are deadly cases. In India, now it has reduced to certain extent. So, an improved understanding of the immunology of malaria is likely to provide key insights into a way to enhance human immunity while reducing disease pathogenesis. So, as I was mentioning, the importance of models. So, patients who died of cerebral malaria, the autopsy samples will give us the terminal event data, all histopathological data or the serological data, and there are issues on ethical issues to study on humans. So, a model can which causes similar severe disease in mice. This is a strain similar to Plasmodium falciparum. This is a strain which causes malaria in this mouse model. It has been used as a falciparum infection, and these are mosquitoes, larvae, and different stages where mice can be infected to the mosquito to recapitulate the way how the infection is happening in the nature. So just to show that to understand the disease mechanism of malaria in human, mouse models have been developed. The mosquitoes are being reared here to infect the mouse, and it will go through liver and then the blood stage where the mice will show fever, coma, and all, so that we will understand, because it's not always easy to do all this basic research in human. So in blood, what is happening? I mean, because when there is cell lysis, infected erythrocyte engulfed by the macrophages, the Th1 CD4 T lymphocytes presented by dendritic cell, it releases interferons, IL-2, IFN gammas, which TNF alpha, which leads to fever. There are splenic red cell removal of the infected RBCs, mitochondrial dysfunction. So all these things are happening in the blood stage, which this studies has been understood via studying in mouse models and several models. And in this picture, we can see that the immune cells in spleen of humans and mouse are almost, this is a human and this is mouse spleen models, and almost similar. There are B cell region, T cell region, the dendritic cells, and the parasite or the pathogens enters in this region, and they are presented by the dendritic cells to the B cells or T cells. So there are research led people to understand that there are similarities among the model system, though there are some dissimilarities also. Not always they convey the same message, as I have explained that depending upon the disease, the model is chosen. So what does the spleen do? I mean, I will say in brief that if your body were a city, the organs and the fluids that make up the lymphatic system would be the policemen, firefighters, and the garbage men. So they protect us. They protect us. The part of the lymphatic system performs number of important functions in the body. They protect us from the outside invaders, stress, and certain deficiencies. So it produces antibodies that fights bacteria and other microbes. It keeps the blood clean, transport fluid and balances fluid levels, remove old worn out red cells. So this is important of one immune organ. There are several aspects of lymph node. The brain plays an important role. The peripheral blood is playing an important role. So during malaria, this is an example of cascade of events happening in mouse model, and how that study helps to translate the research and understand what is going on in the mouse to human brain. So in mouse model, if we see, this is a model which is a sensitive, that they die after the infection of the rodent parasite, that from spleen, the cytokines, the chemokines migrates here. As I mentioned a little bit before, that there is breakdown of blood-brain barrier. So this is a brain parenchyma, and this breakdown is led by the parasite which sequestered here, and the infiltration of the cytokines and chemokines, neutrophils, monocytes, sorry, which leads to the activation of monocyte cytokine secretions, microglial cells, which are brain resident cells, astrocytes. The activation is hampered, which leads to edema, swelling, and finally the mice die. And in a resistant mice, where we apply some therapies or some knockdown mice or some mice where we know that this part is knocked out, like CD8, if it is a responsible factor to cause this events, then in the knocked out mice, like where the CD8 is removed, then there will be no blood-brain barrier breakdown, less parasite sequestration, low patrolling monocytes. Here we can see it's high, and then the resident brain cells will be also in a steady state, and there will be no edema or swelling. So this is an example how immunology and the basic research helps us to understand what is going on underlying the disease mechanism. So I would like to finish here, and before finishing, I want to just say that the immune system is a complex problem, and for every complex problem, there is a simple solution, and it is wrong. So thank you.
Thank you so much, sir, for sharing your valuable knowledge and giving us the right directive to understand basics and recent updates in infection and immunity.
Yeah.
May I request our UG and PG coordinator, Dr. P. Manohar, sir, to share his thoughts on today's webinar. Over to you, sir.
Technical support Dr. K. Munishankar and Mrs. M. Srivani Madam. Other HODs, faculty members of SVDC, anchoring team, student volunteers, all participants, and especially today's resource person, Dr. Tarun Peshwani, postdoctoral scientist in New York. A very good morning to all. This is Dr. Manohar, UG and PG coordinator of Sri Vidyaniketan Degree College. It's my privilege to share my thoughts to this huge gathering of this great international webinar on recent advances in infection and immunity. First of all, we are very much honored to Dr. Tarun sir for accepting our invitation as a resource person for SVDC. I appreciate all the participants who are highly responded and registered to this great webinar. Now, I wanted to say a few words about this today's webinar. The only way to improve human health is to improve human immune system. Decades of work on the principles of fundamental immunity are now bearing fruitful results in the treatment of COVID-19. Several studies demonstrated about autoimmune mechanism for human health. Autoimmune reactions are highly useful in clearing off the excess, unwanted, or aged tissues from the body. Dr. Tarun sir enhanced a lot and brought awareness about infections and immunity among all of us. Dr. Tarun sir has covered fundamentals of immunity, global effort on infections and immunity, coronavirus tracker system. We came to know that no vaccine has approved as of now. Almost all Tarun sir has covered latest developments and current issues in infections and immunity. Dr. Tarun sir has shared the valuable information in a diagrammatic manner. Definitely, this would be the understandable presentation to all of us. I am sure that this webinar helped a lot in a great extent to all the participants. I am confidently says that this webinar has reached the expectations of all the participants. On the behalf of all participants, we thank the resource person for sharing valuable information to all of us. Finally, I congratulate Dr. C. Lekhya Priya Madam and organizers for organizing this beautiful webinar, which is essential for each and every human being on the earth. I hope that this type of webinars are expecting in future also from the Department of Biological Sciences, Sri Vidyaniketan Degree College. Now, I conclude my thoughts with these small quotations. Corona is not much affected the human life as we are thinking, but depression is a dangerous virus. Handling it once builds your immunity to it. In and Buddha says that health is the greatest gift, contentment is the greatest wealth, faithfulness is the best relationship. In this unlock 4.0, I can say maximum possible extent, all of you stay at home and be safe. Thank you one and all for giving this opportunity. Over to Chandana.
Thank you, sir. Now I request Dr. V. Sridevi, ma'am, to start question and answer session. Over to you, ma'am.
Thank you, Chandana. So it was a very excellent presentation session. So now
Thank
some
you. Sorry
questions
for the
from
technical
parties.
glitches here.
Some Rama Mani asked that how to improve our immunity using basic home foods. How to
Using
improve
basic,
our immunity? Basic home foods.
I mean, I think we Indians, I
Like
mean, are more, yeah, means we Indian with our food habits, the traditional food habits, even from childhood, we are immune to several diseases because I'm not going to comment now on COVID, what we are going to eat, which will increase our immunity. But I will say confidently that from childhood, whatever we had eaten, our grandmothers, mothers, we are immune to several diseases automatically compared to the global population. So we are immune. We just need to be strong. And if you see the data which I presented, that we are in a situation where there are very less hazardous cases in India because we are recovering very better than the other countries. So we are immune. Only thing is that we have to be uh, aware that we shouldn't be spreading and maintaining the factor that the social distancing because we are immune. I mean, but otherwise we can have vitamin C's and all which we have in our daily routine. I was audible.
Yes, yes, sir.
Okay.
Sridev, ma'am.
I'm there. Next question, sir, from Preeti Mishra. Is this possible that a person who get care with the homemade remedies can COVID virus again attack on him or her? For example, already recovered from the virus, again they get virus or can attack the virus from or not? There,
It means if I
Preeti
understood
Mishra
the
is asking.
question right, that if a person already got affected before, he or she can get again or not, right?
Yeah,
You're asking this?
yeah,
Yes.
yes.
So there is a case. There is a. Yeah. Can you repeat the question, please?
Which means they recovered from homemade foods, homemade remedies, like turmeric, what you said previously, that the thing.
Yeah, I mean, see, not everybody are
Clove.
turmeric. Yeah, means the question is that if anyone is having homemade remedies, whether they will have COVID or not. You're asking.
Yes, yes, yes.
That part has not been studied yet. The research has not been done, so I can't comment. But I will say that we, if we are consuming turmeric from childhood, we are resistant to several diseases. There are several studies on curcumin, which is the active form of uh, turmeric, curcumin. So there are several diseases where the curcumin is beneficial. It protects. I can't say whether it's for COVID or not, but their studies are going on.
Hello. Next question. How many times a person give plasma?
Can you repeat the question? There is a network problem, I think.
How many times a person can give plasma? Plasma for
How
treatment, sir,
many times
for
a
other
person
person.
can get? Yeah, yeah, yeah. I think 3 to 4
You,
times they can give because there is a time limit also. No, at certain time they have to give that plasma, and also the patients, if they were in severe stage and they recovered, then they have more beneficial plasma. Means it's I'm trying to say that if someone has very mild case and if you compare that plasma with the plasma of the person who are more severe, then the person who was more severe, their plasma they can have in a better way, but it can be taken 4 to 5 times, because after that there will be uh, less antibodies.
Okay, thank you, sir. One more question, sir. So in phase trials, one,
Yeah.
two, three clinical trials, what is the process takes place, sir?
Yeah. What is the process which takes place?
Takes place.
Means you want to know the process in clinical trial.
Yes, sir.
Okay. So initially, first a vaccine candidate is designed based on the research on humans, then in mouse models, in other models. Then after designing that vaccine candidate, it goes to mouse models to understand whether the mouse model is showing the immunogenicity. So immunogenicity means it has to generate, after inducing the antigen or the vaccine candidate, it has to generate the immune response, which can be studied by flow cytometry, ELISA, taking blood from the mouse model, or studying the spleen or the lymph nodes. If we can see that there is a immune response, and then the next step is it should be long lasting. Then after studying in mouse model, it will go to the other model, if possible in human primates, then to patients. So when it will go to patients, then that time the vaccine which was designed for the mouse studies, it should be designed in a way for mass production for human studies. And after the first two phases in human studies, if the results are beneficial, then it goes to the phase 3. In phase 3, if the result is good, then it goes to the phase 4. So here there is one critical part, that while doing the research in mouse models before going to the human in the phase 1, phase 2, there is a lot of paperwork to get the human ethics clearance done, because you can't just randomly give vaccine to human cohorts. There should be a proper human ethics guidelines, and it should be accepted or approved. Then only you can try showing that you have a good results, promising results in your models. Then it will be approved for the human part. Am I clear? Am I audible?
Thank you, sir. Yeah, yes.
Thank you.
Audible. Sir.
Hmm.
Now section is open for question and answers for directly from faculty and students.
Sir, there is another question. Why can't we use bats
Yeah.
as translational models? Why can't we use bats as translational models?
I don't know about that, but I think owls, rabbits, and there are other models which people use for translational models, like owls, people is used for nocturnal animals as they are. But I have no answer to that why bats are not used. I don't know.
Probably you may not get sufficient number, and as you have mentioned, you can have some mice which are knockout mice which can be rare, and probably we can
Yeah.
get the results which are very precise. We may not expect a similar kind of
Yeah.
results if we take bats as experimental models.
Thank you, sir.
Yeah.
Another question is,
Thank you so.
why is that even after being infected with COVID-19, antibodies seem to wane away, but neutralizing bodies seem to long last? Why are memory B cells not generated?
Memory B cells, if are generated, we don't know. We have to study. Because to study the memory B cells, whether they are generated or not, we have to first select the cohort, which the cohort or the patients where they were infected, they recovered, and then if they will again reinfected, then the memory response will come, B cell memory. So now we are not in a stage to know whether the B cell memory response are generated or not. So that's why the scientists who are working on mouse models, who were already working before on the flu or SARS, because suddenly in 6 months it's not possible to establish a whole research in COVID. So those who already had a background set up to understand the virus, or the renowned virologists, they are in verge of understanding with the current newly designed vaccines that whether there will be B cell memory or not. Because I will just add one sentence in the last, that to generate a B cell memory response, you first have to have the primary infection, then after 1 month or 2 months, then the boost. Then after boost, you will be able to detect the B cell memory. So it will take time. I think in a couple of months you will understand.
Dr. Tarun sir, I have a question here. So thank you so much for that excellent presentation. It was very thought-provoking. So I was very much convinced by the last statement of yours that every complex problem doesn't have a simple solution. So I really appreciate that, sir. We need to go for an advanced research and understand. Sir, I have a question, because as a prime time researcher, I would like to bring to your notice that now, as many of us have discussed right now, autoimmune disorders, they have become part and parcel of the society, and the general problems that we have, more stress related or anything, they are having more impact. So in the recent years, especially in India, there was one kind of a one thing that I was very much interested in the presentation was about the resurgence of many other diseases also, sir, as you said, malaria and everything. And India, I would like to bring to your notice that, like, since past 15, 20 years, I've been looking into it. So we were able to see the resurgence of varicella zoster virus, which was part of the chickenpox kind of disease, which was prevalent in the previous days. But nowadays, like, we are seeing that this virus is very prevalent in pre and post pregnancy cases, sir. So sometimes, what's happening is the mother is affected or the baby is affected during the pregnancy time. If the mother is affected, that is one story, and if the baby is affected, it's going to be a generation which is going to get affected with the autoimmune disorders, sir. So it has become one major cause of autoimmune disorders by birth in our country. There has been a resurgence of this issue since past 5 years. So what do you think, how we can actually, do you think the enhancement of the T cell memory is going to help in this case? Because it has been very widely prevalent in India right now.
So if I'm not wrong, you were discussing about chickenpox, right?
Yeah, sir.
So maybe I think there is some sort of mutations or something happened, because chickenpox almost gone away, as you mentioned also. So
Yeah.
I think there is it was not long-lasting memory response. So
Yeah.
maybe that's why, because it was short-lived memory response. So maybe it stayed for several years or decades, but after that, the maybe now people has to be boosted again,
Yes,
so
sir.
that the
Yeah.
memory response comes
Yeah.
again. But
The
it's a
vaccines.
yeah, nice research area. I mean, I'm impressed.
Yes, sir. The vaccines are available, sir, but actually it is not effective. So there might be a
Yeah,
chance where the child will get autoimmune disorders by birth, so which is not very good, because they'll get prone to all sorts
yeah.
of diseases. Yes, sir.
Okay,
Yeah.
okay. So it's a good project to work on. I mean, it will directly
Yes.
impact the
Yeah.
children's. Yeah.
Thank you. Thank you, sir.
Oh,
Thank you, sir.
great, great, madam.
Thank you. Thank you, sir.
How does...
Tarun, sir? Yeah, madam, go on,
Yes,
madam.
sir.
I'll ask after your question.
Please go ahead, sir. Keshav, sir.
Yeah. So, Tarun, sir, why there are no
Yeah.
vaccines for RNA positive strand RNA viruses set? To my knowledge, I think only one virus only has got the vaccine. That is hepatitis C virus.
Yeah.
Other positive strand viruses, even though people are working, but still they are not able to find any vaccine for positive strand RNA viruses. Why is it so?
I will say I have limited knowledge in this aspect, but I would love to get any feedback if someone is here with this answer. But it's a great question, I must say, a thoughtful question. I have limited knowledge in this aspect,
Okay,
seriously.
one more question, sir. Actually, like malarial drug, hydroxychloroquine is used for even coronavirus infections, correct? It,
No,
it,
it was used in a small cohort in
yeah, yeah,
France.
yeah. Maybe because when you were showing like some data, you showed that
Hmm.
cerebral malaria it affects
Yeah.
the central nervous system, correct?
Yeah. Yeah.
And even actually when coronavirus is infecting, the first system that will get affected is central nervous system. Because the
First
people,
lungs.
people who are infected, they normally lose the sense of taste and smell.
Yeah, but
Yeah,
first
right?
the lungs, I think. Anyways, yeah, first the
Yeah. I
lungs will
think
be
one of
affected.
the symptom is loss of taste and smell.
Yeah.
Because central nervous system is also getting affected there also.
Yeah.
Is it why hydroxychloroquine is working for even coronavirus infection?
I mean, I'm not sure whether the hydroxychloroquine is working efficiently on all geographical locations.
Okay.
Means I mean to say it may work to specific ethnic group or population, uh, because in France people had studied that. Scientists had studied that in Marseille. But the mode of action for hydroxychloroquine, which is almost a drug from 1940s, if I'm not wrong, it's still not known in greater detail. Means the mechanism of action, if I'm not wrong.
Okay.
So now it might work in a way that it's killing the host or something. The means where the host cells where the virus is, maybe a better malarial drug now these days, more formulated, may work. I mean, I'm also as if from malaria background, so I have something in my mind that why in African countries and India, which is malaria prone, has lower number of COVID cases. So this is one hypothesis which I have in mind, which is supporting what you are saying.
Okay.
So there is something common there. Maybe, as I said, in India we are prone to several diseases. Many
Yeah.
people already had malaria in India. So I have to just do a basic research. Anyone can do in India that people, how many people who got COVID in India already had malaria before. I
Yeah.
doubt that all of them will have malaria before. Means in
Okay.
contrary, if there are malaria infected people, whether they got COVID again or not in India.
Yeah. Here in India, I think hospital first treatment that they are giving is HCQ for first 2 days. After
Yeah,
admitting into hospitals, the first
yeah,
month
but it
what
has.
they are doing is actually 800 milligrams HCQ for the first day, and second
Hmm.
day I think 400 milligrams. So that
Yeah,
is what
means
the
if
treatment they are starting with in India. So that is why I feel like maybe it has got something to do with the central nervous system.
if
HCQ. I
it
don't know.
does, then it will be a great thing actually. I mean, already in USA there was a huge supply from India for the hydroxychloroquine 2 3 months back. But here patients showed side effects actually.
Okay.
So that's why I'm saying it depends on the, I mean, geographical location also sometimes. I mean,
Yeah. Thank you. Thank
because
you, sir.
that's, yeah. But it's a good thought process. I mean, I'm impressed.
Thank you, sir.
Thank you, sir.
Thank you.
There is another question, sir. How does herd immunity play an important role, like in current scenario, especially when a vaccine isn't developed yet? Can a person who acquired herd immunity alter results in the clinical trials?
Can you repeat the last few words?
Can a person who acquired herd immunity alter results in the clinical trials?
It's a good question. So, a person acquired herd immunity alters research in clinical trial. No, because first of all, to gain herd immunity, it should be gained by 70 to 90% of the population. You can hear me?
Yes, yes, sir.
Yeah, and so when researchers are planning the cohort studies, when these days I have seen my boss also here working on COVID patients, so they are very meticulous and precise to design their cohort. They will not design a cohort on the basis that, suppose they will have the information that that population or that locality or that region people already went to herd immunity, so that population will not be included, or that individual will not be included in the research study, so that it will make the outcome biased. So it's a very good thoughtful question. I would love to know who asked this question, but it's a very good question. But normally they will not be included, I mean, if that population or community are in herd.
Yes, sir. Thank you, sir. why the infection is more prone in males than compared to females. Does estrogen has influence on less prone of infection to COVID-19?
I have no idea because this I can't comment. I didn't saw that work or the article where it has been published. But I can say in malaria, there are some bias that females are more impacted with the falciparum and children also get males are less. So, but I don't know this part.
Thank you, sir. The another question is how to boost up our mental health during this pandemic.
How to boost the mental health? Yeah, to first thing is that to understand with the see we can see the news in a way that it's positive that there are less more recovery and less deaths. But the thing is that we are seeing the news in a way means I'm saying news. Why? Because it's the only thing which is coming popping up in front of you every day. So first thing is to avoid that, and the second thing is to discuss with your friends, speak, and this is the best time to think that in last 4 5 months we didn't went to the diagnostic centers, didn't visited the nursing homes, didn't went to our family doctors. That means we were always fit and healthy, but it was our psychological stress before the pandemic. We were visiting doctor every 1 month. Everyone has a family doctor, so basically COVID made us more fit and healthy.
Yes, sir.
Very
Uh,
true.
Sir, shall I ask a question for you, sir?
Yeah.
So, what is the role of vitamin C in COVID patients? Means doctors are saying to take more vitamin C and all, no, sir. What's the role?
I mean, if doctors are saying not to take vitamin C, then someone shouldn't be taking vitamin C. But if someone is asking that what boosts our immunity, then I will say vitamin C. Means you have to understand that if you are not immunologically strong, then you are prone to many things like influenza, flu, and other aspects. So that which corroborates with the fact that also comes corona. So, I mean, doctors are saying maybe in this case, I don't know, uh, but it's quite a healthy way to be fit and have vitamin C, D's, and we have a lot of vitamin D's, but vitamin A. On the other hand, there are multivitamin capsules and all, but if people is fit and fine, having good food, it's okay. But with stress, people are more prone to any disease.
Thank you, sir.
Yeah.
Good,
Yes, sir.
Chandana.
Thank you.
People who show no symptoms of COVID-19 are having antibodies when tested. Why and how, sir?
Can you repeat the question, please?
People who show no symptoms of COVID-19 are having antibodies when tested. Why and how, sir?
People who don't have symptoms of,
Symptoms. COVID-19 are having antibodies when tested. Why and how, sir?
I mean, I might say with my limited knowledge that sometimes the antibody test is not accurate. But I don't have the exact knowledge or idea in this time point that why they are getting the antibodies. But even if they get the antibodies, the titer will be very low. So it can be two way. If the antibody testing is very sensitive, then that's why it's able to detect the lower titer. Or otherwise, the antibody testing sometimes is going on the wrong way. Uh, Otherwise, there is no scientific conclusion of this part. I mean,
Thank you, sir.
think-
Dear students, could you please interact with our resource person to clarify your doubts?
Sir, I would like to ask one more question. For the students to take research products at the graduate level, so what are the areas that you can suggest? Looking at the Indian scenario, we have the microbiology department here with us, along with the Department of Chemical Sciences. So I would request you to suggest few project areas for the students so that they can do, because we do have a research center here. So I hope that with your motivation and guidance, they will be able to take a few projects in the coming days.
I will suggest, I mean, the students to first understand or shadow, or you can send students to institutes. You can ask in research institutes that your students will go there. They will shadow the other researchers there. They will just see what's happening, and from there, with 1 or 2 months experience, they will visit various labs. They will not touch anything, but from there, let them gain some idea, and then ask them, now what do you think? Because that will help them to understand that what's the research setup. Like,
Yeah.
means if we decide now for them, so they will not get an open mind. And I
Yes.
want them also to see that what they are doing with the pipettes, what animal model is, what research they are doing. So seeing a basic or translational research, even if you can send them to nursing homes and hospitals to see what and how a nursing home or hospital is doing with the research unit, because there are always clinic and research collaborations, so they can go in both way. On the other hand, if someone is interested in bioinformatics or computational skills, then he or she can also go to some labs where they do bioinformatics, and they can just go there for 1 or 2 months internship. They will gain some knowledge, and then they decide a project for themselves. So I think the initial thing is their inspiration and motivation, which I am sure with the organizational skills which your institute has, they will definitely are in good hands, I will say.
Thank you, sir. Thank you so much.
Dear students, you could raise your hand so that we will unmute yourself to clarify your doubts. Yes, Tejas.
Hello.
Yes, Tejas.
Hi. Hi, Tejish.
Good morning, sir. Good morning, sir. This is
Good
Tejas
morning.
Kumar from second year MBC. Sir, is
Yeah, I
there
can
any
hear you.
possibilities to cure coronavirus by using stem cells therapy?
By using stem cell therapy, are there any treatment you are asking, right?
No, sir. Stem cell therapy.
Yeah, stem cell therapy is treatment for corona, you're asking me.
Yes, sir.
Till date, no studies are out yet. So for sure, definitely. I mean, people are working on that, but I doubt whether with the viral infections, stem cell therapies will work. So vaccines will work, I think. I doubt.
Okay, sir. Thank you, sir.
Thank you.
Shiva Gangadhar.
Good morning,
Sir,
sir. Good morning.
Good morning.
Can
second
mosquitoes
language.
act as a transmitters for COVID-19, sir?
Yeah, can you repeat the question, Siva?
Can mosquitoes act as a
Yeah.
transmitters of COVID-19, sir?
No, 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 no,
After biting the infected person,
no, no, no,
and
no, no, no. Because the mosquito has to carry or able to reproduce the viral part. It has to be a carrier. It can't. It can't.
Pushpalata.
Second BSc. Sir, does hypertension suppression? If it is, because why, sir?
I can't hear you. Hypertension, then after?
Does it cause immunosuppression? If it is, because why?
Hypertension do cause immunosuppression, but to what extent that need to be understood, that how prolonged the person is going through hypertension. Because it will, because prolonged hypertension may lead to cardiac arrest or other issues which may cause the immunosuppression, not only the hypertension. So there should be something related to that, and the prolonged hypertension may lead to something else.
Is there any other questions from the students?
Sir, good morning, sir.
Good morning.
Sir, this is Vijay Bharatrandir from 2nd Microbiology Department, 2nd year.
Hi, Vijay.
Yes, sir. Sir, I have a doubt that COVID-19 strain can be changed according to the environmental conditions and climatic changes. That we can see that it is originated in China, that it is evolved in China, but China and other countries having different climatic variations. But even in that conditions, how it is able to survive and infecting lot more people than China?
So, I mean, you already answered your question because you said that the different countries, so different countries, if the COVID strain is different, the host is also different, right?
Yes,
Means
sir.
sitting here in USA, if someone is getting infected with suppose a similar COVID strain in India, after several transmission, the host is also different. So it will change and it will adapt according to host, because if a virus is not adapting to the host, because it needs a host where it can replicate properly. So you already answered your question yourself.
Okay, sir. Thank you, sir.
Thank you.
Sir,
Yeah.
sir, I would like to ask one more question, sir.
Yeah, sure. Go ahead.
Sir, can cloth mask can stop the coronavirus, sir? Or which mask would you like to suggest, sir?
See, any mask will stop the spread of coronavirus because it's transmitted from human to human by respiration. So any mask will stop the transmission, and also, I mean, not only wearing mask, maintaining a distance is also important. But while keeping a mask, people are speaking closely, like not 6 feet, but lesser than that. But any mask will work.
Thank you, sir. And coming to the sanitizers which we use, sir, really shows the
Yeah.
effect on coronavirus, sir.
I mean, it will not show the effect on coronavirus, but it will show the effect of sanitization. Like if you are sanitizing your hand, you should be free from germs, any germs. And as there, we know that the coronavirus stays on the metals and the clothes and the other things when we travel or somewhere. So I think this practice should continue now for coming several decades, so that if there is any other outbreak coming in future, that time it will be less spreaded, like they do in Japan. I mean, they're always
Yes. Thank you, sir.
wearing gloves. Yeah. Thank you.
Is there any questions from the students? If you have, please raise your hand.
Sir, good morning, sir. This is
Good
Mahesh
morning.
from First Embassy, sir. I have
Hi,
a small
Mahesh.
question. Sir, do pregnancy ladies suffering from COVID, is COVID virus transmitted to the fetus, sir?
There has been some studies on this, but no positive cases has been found till date. Means people had studied COVID positive pregnant women, but when the child was born, the COVID was not transmitted. So the studies has been done on few people. So there are no cases yet.
Okay, thank you, sir.
Okay, sir. Thank you so much, sir, for your patience in answering all the questions. Thank you, sir.
I tried to answer. I mean, I didn't knew all the answers, so I tried. But they were very nice questions from everyone.
Thank you, sir.
Madam
Over to
Katyani wants to ask.
Katyani wants to, yeah.
Katyayani. Katyayani, unmute yourself now. I have given permission. Yes.
Good morning, ma'am. Good morning, sir.
Good morning.
I am Katyani Naidu and I am in the final year of my graduation.
Good.
And my question is, why is COVID considered so dangerous? I mean, like, what does it actually do to the lungs that it is considered so dangerous?
So for lungs, it damages the lungs, and that's why it leads to the respiratory distress. There is a respiratory problem, which leads to the reduced supply of blood in the other organs, which leads to multiple organ failure. and systemic death. So, if the lungs are unable to recover from that stress, then patient needs to be in ventilation. And after that, if they are unable to recover, then they die. So, COVID in the severe people, they do a lot of stuffs with the comorbidities, the term which is coming up that if they have something else also. But otherwise, your question in the beginning was okay that why COVID people are scared of. I mean, the people who are scared of who has something serious. Otherwise, people are scared of spreading to other people. So people are aware. That's why they are scared.
Okay, sir. Thank you, sir.
Thank you.
Thank you, sir. Over to
Thank
Sai
you.
Chindana.
Thank you, ma'am. Thank you, sir. I request our head Department of Biological Sciences, Dr. S.V. Ravikant, sir, to propose vote of thanks. Over to you, sir.
On behalf of the organizing committee, I would like to thank Dr. Tarun, postdoctoral scientist, for his excellent talk. I would like to inform the student community that he is working in Albert Einstein College of Medicine, which means he is working in a place which is probably related to hospital. And most of the students from biological sciences, they have this kind of interest. They want to get into medical college. So if you can pursue research, you can work hard, you can definitely get into a medical college where you can definitely do your research. That is the reason I emphasized the word College of Medicine. Dr. Tarun, this is just the beginning. We would like to associate ourselves with you in future, and I'm pretty sure some of our students will explore the opportunity pertaining to immunity, research pertaining to immunity. Dr. Tarun has mentioned that all the vaccines may not provide 100% efficacy in curing or efficacy in treating the diseases. Therefore, there is always scope for research wherein you can go for a better vaccine. So that is the reason I told you that this is one of the areas which is definitely a hot cake in research. As students of biological sciences, you can definitely choose immunology as your career and find solutions to several diseases. We all know about malaria. Malaria is not a new word. When I was in intermediate, also I studied and also studied about the treatment to malaria. But even now, people are doing research in malaria. That means that you have different strains of malarial parasites, or perhaps the cure that we felt was fine is not complete. So this indicates that research pertaining to immunology is always a hotcake. The kind of trauma the whole world is undergoing due to this pandemic is hard to put it in words. I am sure people like Dr. Tarun will soon bring solace to all of us. My sincere thanks to our beloved and dynamic principal, Dr. Asha Lata, for her support and encouragement. I'd like to just mention a point or two. Some of our students know that she is from English department. The kind of questions she was asking, she was talking about autoimmunity, she was talking about B cells, she was talking about various diseases. That indicates that you need to have a lot of passion to read. We should not restrict ourselves to our area. Don't just tell me that I belong to microbiology. Now you have to have a blended research or an interdisciplinary research. And I want all the student community, especially, to know that I have to think out of the box so that tomorrow I may find a solution to a particular problem. Thanks, madam, for all the encouragement and support. I would also like to thank our UG and PG coordinator, Dr. P. Manohar. Again, he belongs to statistics department. He did sufficient homework and he could speak some sentences in immunity. Understandable had he belonged to biological sciences, but a person who is from statistics background, who has studied mathematics, statistics throughout his career, if he is able to understand and speak something related to immunity, obviously that is definitely a very good sign. And we can take a leaf out of their books, and we can also read subjects which actually do not belong to us. Okay, thank you, Dr. Manohar, for all your support. On behalf of the organizing committee, I would like to thank Dr. Keshavadu. He is one man who always keeps the webinars alive, and uh, he is an excellent researcher, worked as a postdoctoral fellow in foreign countries for over 8 to 10 years, and absolutely down to earth. We are extremely happy and we consider ourselves extremely fortunate to have him in our team. Uh, Thank you, Dr. Keshan.
Thank you, sir.
I would like to express my thanks to Mr. Munishankar and the computer faculty for their technical support. They were responsible in designing the feedback forms, Google Forms, and all that. I would like to convey my thanks on behalf of my department and on behalf of the organizing committee as well. I would like to express my heartfelt thanks to all the participants and students without whom this event would not have been unfolded. I would like to thank student coordinators, Ms. Sai Chandana, Swati Ramchandran, Mr. Mahesh, and all others. And they have been instrumental in helping our webinar coordinator, Dr. Lekhya Priya, from time to time, and making this webinar a grand success. God bless you all. I thank our management for supporting us in all possible ways and making this event possible. I would like to thank the media team of Sri Vidyaniketan Educational Trust for designing brochures and e-certificates. I would like to place on record that the webinar coordinator, Dr. Lekhya Priya, for taking the complete responsibility in organizing the webinar. She has kept herself extremely busy for the past 2 months, and whenever she organized a webinar, more than 90% of the work she alone did it. And that has really lessened my burden as head of the department. And I would like to definitely thank her for the energy that she has displayed. I am pretty sure she's again ready for the webinar, which we are planning in the month of, we are planning in a couple of weeks ahead. She already told me that, sir, give me the related works and I will start doing it. Thank you, madam, for all your uh, help and organizing, because at the end of the day, whatever credits I get it as head of the department, I should definitely tell you that it is because people of the potential of Lekhya Priyas have contributed a lot. Thank you, madam. And the, I would also like to thank our colleague Dr. Sri Devi. She had a lot of network problems. In spite of that, she could take some time off and note down all the questions and try to convey it. It is very difficult to uh, get good network in the area that she is residing. In spite of that, she could make it convenient and she could note down all the questions from the participants and convey it to the resource person. The feedback link is shared through WhatsApp group. I request you to fill and send the feedback forms, and e-certificates will be issued to you all. Immunity always depends on the response. So if you look at this lecture, it was almost 1 hour lecture and almost half an hour response from the students. That indicates that this was a huge success. And very patiently, he has answered all the questions. Dr. Tarun, he looks pretty young, and I am sure in years to come, he will do lot of wonders. God bless you, Dr. Tarun, and
Thank you,
back
sir.
to you, Chandana.
Thank you, sir.
Thank you.
I thank all the participants for joining us today. Kindly fill the feedback forms for receiving your e-certificates. Best wishes, take care. Over to Lekhya Priya, ma'am.
Thank you, Sai Chandana. On behalf of Department of Biological Sciences, I am happy to express my sincere gratitude to our Honorable Chairman, Dr. M. Mohan Babu sir, our revered CEO, Mr. Vishnu Manchu sir, for giving us an opportunity to organize this event. I am very grateful to our principal, Dr. D. Ashalata ma'am, for providing guidance and encouragement. I would like to thank our beloved HOD, the Department of Biological Sciences, Dr. S.V. Ravikant sir, and our UG and PG coordinator, Dr. P. Manohar sir, for their constant support. We are extremely thankful to our resource person, Dr. Tarun Keshwani sir, for accepting our invitation in his busy schedule and sharing his valuable thoughts and experiences with all of us. Thank you, sir. We have participants from all over the globe. We thank all the participants without whom the event wouldn't be successful. As no program can become successful with a single person, so I extend my big thanks to the organizing committee members, Dr. V. Sridevi ma'am, Mrs. M. Srivani ma'am, and Mr. K. Munishankar sir, and all our student volunteers, Ms. Sai Chandana Venkatila, Ms. Swati Ramachandran, Mr. Vijay Bharat Ranadeep, and Mr. P. Mahesh. Thank you for making this event successful with your contribution. Since registered participants are more than 700, we request you all to submit the feedback before 2:30 p.m. today. Once again, I am repeating, since registered participants are more than 700, we request you all to submit the feedback before 2:30 p.m. today. Feedback link is displayed in YouTube chat box. Feedback link is displayed in YouTube chat box. Those participants who fill and submit the feedback receives e-certificate. Take care, be safe. Thank you one and all. Over to Principal
Thank you.
ma'am. Thank you, sir. Thank you so much.
Much for joining us. Hope God will bless all of us to come out with wonderful research to make, as Ravikant sir said, to bring solace to the world. So we are all there together. As you said, pandemic has made us more stronger. So we hope that this is going to help us to become much more dynamic in the coming days, making ourselves immune and also boost up our immunity, take up new, dimensions of research as well. Thank you so much. Thank you so much.
Thank you. Thank you, ma'am. Thank you, everyone.
Thank you, sir. Thank you, sir. Thank you, one and all.
Thank you. So I'm leaving. It's okay.
Yes, yes, sir. Thank you, sir.
Take care.
Thank you. Thank you.